Thank you. Thank you for coming to our presentation for Benedictine colleges, advancing the new evangelization technology in the human person. Fritz Kahn was an enthusiast, an early enthusiast of man-machine comparisons, particularly that of a chemical plant, comparing the body to a chemical plant. This was at a time that the German chemical industry was the world's most advanced. Our aim is that by the time we're done, you'll have a new appreciation for the latent disaster in this phrase, man as industrial palace. The irony is that multiple lines of evidence are converging to link that disaster to an actual physical product of mid-20th century German chemistry. Most of you probably come from a humanities background. I suspect most of the talks during this symposium are also from a humanities background. Dr. Sprouse and I do not come from that background. We come from a biological science background. And so we expect that we'll have information that may be new to you, that will perhaps offer an alternate explanation for some of the things we're seeing in society right now. One of the goals of this conference, the stated goal of this conference, was to turn an eye toward the ways in which technological society is changing our understanding of the human person. And a thesis of our presentation is that technology is not just changing our understanding of the human person, it's actually changing man himself. And in an even more concrete way, not so much mankind as an abstraction, but men. Technology is changing men. So how are men changing? Men are changing in four ways. They're changing in fertility, they're changing in virility, changing in anatomy, and changing in behavior. And a sampling of these changes that are going on are seen in today's headlines, where the rapid succession of one change after another, all with a common underlying theme, are sort of like a runaway train. Prompting the subtitle for our talk, Zwitterbahn. Zwitterbahn is a German neologism, and stemming from the root word Zwitter, which was originally used to describe the charges on an amino acid. And at body pH, amino acids have both positive and negative charge. They share the properties of two opposing poles. And the German word Zwitter literally means hermaphrodite. Fertility. This article was in September's GQ magazine, and it was a popular distillation of some of the problems that are happening in society today. And one of the phrases in the opening paragraph was, men are doomed. And certainly that smacks a little bit of sensationalism, but it referenced this article by the Danish andrology expert Skakkebak. And Skakkebak and colleagues presented a series of problems in society in a scholarly biomedical journal, among which was dropping fertility in young men, and he used the actual phrase tipping point. 20 to 30% of young men today have a sperm concentration below 40 million, which is associated with reduced fertility. And this is another author, Haggai Levine from Hebrew University and Mount Sinai University in New York. He and authors published a meta-analysis, which is a rigorous statistical compilation of other studies on fertility in men. And I would, at this time, point your attention to the major curve there, the black line, which shows that in the last 40 years, the decline in fertility counts. This is new data. This is relatively new data. The Endocrine Society, which is a collection of experts in hormones, published a survey of problems with male hormones just 3 years earlier, and this was not in there. At that time, they said, hey, the jury's out. We don't really know if counts are decreasing, so this is the most recent information. One textbook published last year said that, at least in the U.S., in 1940, the average sperm count was 118 million. And in 1990 was 66 million, which is just about half that of the previous. So that would agree with this data. And the data comes from multiple studies. These studies were in 50 different countries. And you can see the individual points. The circle is sort of a bubble plot here, and then the line down the middle is the average trend downward. This is another way to look at the same data. So this is from Denmark, the last 15 years. And the bars are the number of infertility procedures in Denmark by year. And you see the trend is going upward. So increasing number of infertility procedures speaks to infertility. At the same time, the dotted line is plotted on the same graph. The dotted line is the number of women of reproductive age. So you can see that while the population of people who are actually trying to get pregnant is dropping, there's an increasing rate of infertility procedures. So the rate of infertility must be rising even more than the bars would say. And there's a personal impact of this. Childlessness. There was a very large study of Danish men involving more than 1 million men born between 1945 and 1980. The early cohorts, the early baby boomers, there was a 15% rate of childlessness. But by the time it got to Gen Xers, there was a 22% rate of childlessness. And the preceding graphs would allow us to look beyond the common assumption today, at least in academic settings such as Benedictine University, that's just decreased desire. People aren't wanting to have kids. People are delaying kids. But you can see this is occurring in the context of decreasing sperm counts. And these counts also have a societal impact. So what you see here is a hypothetical projection forward of what would happen to population at certain fertility rates. The dark line is the current fertility rate in Western Europe. So right now they're having 1.5 children per family. So if that continues forward for 3 generations and there's no immigration, in 3 generations there will be 60% fewer people in Western Europe. Virility. This article from Forbes magazine, obviously an attention-getting headline, speaks to a decrease in testosterone levels. So I think the common assumption today, there's a lot of advertisement of testosterone supplements, and you hear it all the time in sports radio. So I think our common assumption is that, well, this is just an oversexed society. This is, old men trying to relive their youth, or perhaps misusing medication. But the author here speaks to something on the next slide, which is that underneath it all, there's actually something real, something physical. This is U.S. data involving 1,300 men. Some of them measured 3 times over 18 years. But what you see is that as you start with people born about the time of World War I, they had the highest testosterone levels of all. People born during the Great Depression, a little lower. People born during World War II, even lower than that. So there's a trend over time. The later you're born, the lower will be your testosterone level, at least in the United States. Leading to the conclusion in this professional hormone publication, calling it a generational decline in testosterone levels. And this might have a role in something that's been attributed entirely to social phenomena, which speaks to perhaps a drive to succeed. And this shows that the breakdown of college degrees awarded by gender in the United States, and it used to be kind of unequal, with 60% going to males and 40% to females around 40 years ago. And now the trend is reversed. And so, a lot of this is good, and I think that's what we all knew about, that nowadays there's increased opportunities for women, more attention being paid to the educational style of women. But there is some data, particularly offered by the proponents of this book, that there's also a creeping deficiency in male performance, and perhaps it's due to drive. For example, 70% of valedictorians in the United States are female now. 70% of those who get D's and F's are male. Boy IQs have dropped 15 points since the 1980s. And men 25 to 31 years of age are 66% more likely than their female counterparts to be living with their parents. Dr. Farrell was one of the original board members of the National Organization for Women, and Dr. Gray is the author of Men Are From Mars, Women Are From Venus. It's a good book. Anatomy. So cryptorchidism is a medical word for hidden testicles, something that's been known for a long time to physicians. So, of course, the testicles are supposed to be inside the scrotum. It's a good place for them to be. They work better when they're out there. There's less risk to the cells when they're at a cooler temperature in that location. But cryptorchidism is when people are born with the testicles in the wrong location. They're inside the abdomen, or perhaps in the inguinal canal, which is where hernias occur. And this, too, has been rising over the last 50 years, 60 years. This data shows two different countries, Denmark and the UK, and the increasing rate of cryptorchidism. The Endocrine Society says that in the late 1950s, the rate was 2%. However, in the 2000s, it was 8%, so it went up fourfold. Now, cryptorchidism was, and still is, classically understood to occur at birth. If you're born with the testicles in the scrotum, they'll be okay the rest of your life. Some people aren't born that way and it has to be fixed. But I remember the first time I encountered this in my own medical practice was I was sure I had documented that the patient's testicles were in the right spot. And yet, sure enough, when I was checking them for an athletic physical later, when he was a young adolescent, they weren't there anymore. They had basically hidden. And I didn't quite know how to explain it. Sometimes when we see strange things as a physician, we offer sort of a hand-waving explanation until it happened again. And that time, I knew I'd seen it before and started to think there's something real about this. Still didn't understand it until I was researching this talk, and I found out it is a known phenomenon, but only in the last 25 years. When I went to medical school, no one was taught about this. I recently spoke to two of my colleagues about this. They were unaware it happens. And since my second patient, I found a third in the last 4 months. All things I never knew of in med school. It's not like this is a diagnosis that requires advanced technology and MRI, something that wasn't available 25, 30 years ago. It's a very simple thing. People have known how, doctors have known how to examine this for hundreds of years. But it's only been a reported phenomenon for the last 25, which makes you think that perhaps it's a new thing. The reason we didn't know about it before is it didn't occur. And so in this phenomenon, the testicles at birth are in the right place, but later on they just go back up inside the abdomen. Another anatomical change is an increasing rate of hypospadias. This is not a plural word, it's actually a Greek word. And in this condition, what happens is the normal opening of the urethra where the urine comes out on a male is moved from the tip of the penis to on the underside of the penis. And this too has been increasing in multiple countries. So cryptorchidism, hidden testicles, and hypospadias, they have more than one thing in common. They've both been increasing. They're both alterations in male anatomy. They have something else in common. And that is that they are a change on the spectrum, on the continuum, going from more male to more female. In the case of the external male genitalia, hypospadias, so this is the feet, this is the early embryo, the structure of the organs that are going to develop in the external genitalia. And you see female on the left, male on the right. So the opening of the urethra is supposed to be here, but in hypospadias it's down here, which is analogous to what happens in females, where the opening of the urethra is behind the clitoris. So this is the case where, at least in the external genitalia, males are becoming more like females. And in the case of internal genitalia, the gonads, here's the early embryo where the gonads are inside, and then in the males, they're supposed to move out here, and in the female, they're supposed to stay inside the abdomen, because the ovary is the analog of the testicle, or vice versa. But when you have cryptorchidism, the testicles stay up inside like an ovary. Lastly, in terms of anatomy, there is very small but measurable change in sex ratio. This is happening in more countries than not, where the ratio of males to females at birth is going down, and that's a great thing. Of course, the genders are equal, but there is a change all the same, and it makes you wonder why, particularly in the context of these other findings. Lastly, behavior. Data on this is hard to find. It seems to be something that is not perhaps popular to measure, but it is an important social phenomenon, and we should probably measure important social phenomena. And this data is from the Pew Research Center, which I think is a pretty reputable organization, and they used a Gallup poll over the last 6 years to determine that the rate of homosexuality is rising. Alongside that, it's what we've also seen in numerous articles in the press, and I think many are concerned about at places like Benedictine, and that is the increase in transgender ideology and gender dysphoria. So, the components of the male endocrine crisis include fertility, virility, anatomy, and associated with that, not included in the male crisis by most authors, are parallel changes in behavior. All these sharing a movement from male to more female. Now, what does this have to do with technology, the topic of this conference? Well, the male endocrine crisis appears to be environmental. It predominates in Western countries. And this is the case for sperm count. So this same graph I showed you earlier. So the black line is basically all men in Western countries, such as Western Europe, United States, New Zealand, Australia. This tan line is men in other parts of the world, such as Asia, South America, Africa. And this line from the West is statistically significant. This line from the third world is not. So, you know how statistics are, something's real if it's statistically significant. So even though it appears to slant downward, it was not a statistically significant finding. And the other two lines, this is men in the West who are fertile. That line is still going down. And then this is the line of men in the third world who are fertile, and that's actually going up. So there is definitely an east-west split between the technological countries and the non-technological countries in terms of fertility. At this point, I'd like to break and talk about another phenomenon called the testicular dysgenesis syndrome. This is actually advanced by Dr. Skakabach, the expert in andrology from Denmark. And the testicular dysgenesis syndrome consists of precancerous cells in the testicle. And it's associated with, guess what? Low sperm count, hidden testicles, and hypospadias. Sound familiar? And the precancerous cells are also associated with a rise in the rate of testicular cancer. Now this too has an east-west split. So in this graph, you'll see that there are 6 regions of the world: Northern Europe, the Americas, Asia, Eastern, Southern, and Western Europe. So you see in all areas it's increasing, but there's a dichotomy between more developed countries and less developed countries. So for example, in Northern Europe, you have Norway way up here, and you have Lithuania down here. In the Americas, you have the USA whites up here, Colombia down here. In Asia, you have New Zealand up here, Philippines, low rates. And in Eastern Europe, Czech Republic, high rates, Belarus, low rates. Southern Europe, Slovenia, high, Spain, low. And in Western Europe, just about every country has a high increasing rate of testicular cancer. Now, only 25% or less of testicular cancer is explained by known genetic variants, which is another way of pointing to a suspected environmental cause. So, another factor that suggests environmental role for increased testicular cancer that's associated with these other changes, so the male hormone crisis, are that men who move from countries with a low rate of testicular cancer to a country with a high rate of testicular cancer, their sons, born in the developed world, will acquire the risk of the host country. So even though they've got the same genes as the people in the old country, now that they're living in the developed West, those kids conceived and born there are going to have a higher rate of testicular cancer. Same genes, different risk. And the other thing is there appears to be a window of development, a window of time in which this risk is acquired. So if you're a young man moving from, say, Philippines to New Zealand, it's not going to affect you. But when you have kids in the new country, the West, your kids conceived and born in the new country will develop the risk in the new country, suggesting that there is a window of vulnerability in the womb. So evidence for environmental cause for the male endocrine crisis. Fertility is decreased in the West, not elsewhere. Virility is low in modern, not previous times. High testicular cancer rate is acquired by moving to the West. That also is a modern risk. And the high testicular cancer risk is associated with the above-mentioned changes in male anatomy. So, at this point, moving to a discussion of potential environmental causes, I'd like to have Dr. Sprouse.
Thank you. So, while these alarming changes are not entirely clear, we do know that just as the canary was long used as a sentinel to toxic gases in the coal mines, there are some very disturbing changes happening in fish that may serve as a sort of warning to us and give us a clue as to what environmental problems we're facing. Our awareness of these changes in fish dates back over 20 years when hermaphroditic fish were observed in sewage treatment effluent in England. Now, this group decided to design a study where they placed caged male trout in the effluent, so the water leaving sewage treatment plants. And what they found was that in these male fish, there was an increase in this egg yolk protein, which was at levels characteristic of ovulating females. What they did next was they placed trout in glass aquaria with ethylene estradiol, and they saw comparable levels of increase in this egg protein. Now, ethylene estradiol is the synthetic estrogen in contraceptive pills. The next breakthrough came in about 1998 when a study was published where they looked at separating the components of the effluent from 7 different sewage treatment plants using a series of chromatographic separations, and the fractions of water that were activating the estrogen receptor were analyzed with mass spectrometry. And what they found was that 3 steroids and no other chemicals were identified in these active fractions of water. They were the natural hormones estradiol and estrone, and the synthetic hormone ethylene estradiol, or EE2 as I'll call it. They checked their findings by spiking some of the samples with EE2, and then they found that the fractions separated into the same fractions as those active components of the water. EE2 is the estrogen component of almost all modern formulations of combined oral contraceptive pills. It's almost identical to estradiol, the natural hormone, except with the addition of this acetylene group, which makes it more bioavailable as well as more resistant to metabolism. It is a lot harder to degrade, which makes it a more effective contraceptive, but as we'll see, it's not so good for the environment. So how does this end up in our water? Well, the average daily dose of EE2 for a woman taking contraceptive pills is 26 micrograms per day. And of that dose, only about 43% is fully metabolized in the body. The rest is either excreted in the feces, where most of it is the parent active compound, or in the urine, where some of it is conjugated in an inactive form, and some of it is readily deconjugated back into the active form. And so the total of the original dose, about 40% of that that's ingested, is released into the environment, and that's about 10.5 micrograms per day. So once it's excreted, it enters into the wastewater, which then goes on to sewage treatment systems, which vary in their treatment of the water, but in general, the water enters into the sewage treatment plant, and that's called the influent. It passes through a series of screens to remove larger objects, then goes on to a grit chamber to remove things like gravel, dirt, kind of bigger particles like eggshells or seeds. Then it goes on to a sedimentation tank where the heavier solids, called the sludge, will settle to the bottom, and the greases and oils are skimmed off the top. Usually it then goes on to a secondary treatment process where microbial decomposition of organic contaminants will further purify the water. Then the water leaving the sewage treatment plant is called the effluent. And there was a removal rate cited for EE2 that was based on 37 different sewage treatment plants, measurements of EE2 at these plants, and the removal rate was 52% for EE2, and they said it was highly variable between plants and within plants, depending on sort of the temperature and different conditions. But on average, if about half of the EE2 is removed during that treatment process, then half of it is going on into the environment. So upon leaving the sewage treatment plant, the effluent then enters surface waters, lakes and rivers, and since EE2 is a fairly hydrophobic molecule, it likely accumulates in the sediment, and that could act sort of as a sink to further contaminate the water long term. The EE2 is also in the sludge, which is commonly used as a fertilizer on the soil, and so there's the potential for the EE2 to leach into the groundwater as well. This is a brand of fertilizer that is made from the waste, the sludge of Milwaukee sewage treatment plants, and has been since 1925, and thousands of tons of it are sold per year. So once it reaches our water sources, what is it doing to aquatic organisms? We will see from many controlled laboratory studies that it is causing problems that are very similar in fish that we're seeing in the male endocrine crisis that Dr. Herrick outlined. Now, this isn't proof that contraceptives are causing the problems we see in men, but the comparisons, the similarity certainly suggests that someone raised the question. So fertility. EE2 has been tested in a long-term study on, what was this one? The fathead minnow in an experimental lake, and even at low concentrations, it caused many problems and actually near collapse of the species. Another study shows that also low concentrations caused this elevated egg protein and feminization of all of the fish exposed, in this case zebrafish, and inhibited their reproduction. Even short-term exposure of the zebrafish reduced the gene expression of genes that are responsible for male sex determination. Another study looked at the sperm quality of tench after exposure to EE2 and found that all measures of sperm quality were reduced. Virility. So in addition to the effects on fertility, there are also effects on sort of the male typical behaviors of certain fish. So after exposure to EE2, even at small levels, the zebrafish showed a reduction in aggressive behavior and courtship behavior, and that likely contributes to some of the reproductive problems. The pipefish had their mating dynamics disturbed. In particular, the males were less attractive to the females after being exposed to EE2. The zebrafish also were analyzed in terms of their anxiety and shoaling behavior, and the EE2 tended to just alter those behaviors and make them deviate from normal. And since these are adaptive behaviors, it likely affects the fitness of the species and the reproductive success. Anatomy. So numerous studies show that EE2 causes feminization of male fish of many species. they either develop ovaries, they don't properly develop their male gonads, or they display female secondary sex characteristics. And even at low concentrations, what's commonly observed and measured in the environment, this is happening to zebrafish, where they see complete sex reversals and suppression of gene expression that is responsible for forming male gonads. And this doesn't appear to be just limited to fish. There have been studies that EE2 causes feminization of frogs and disrupts the genital development of rats. Now, considering the abundant literature on the toxic effects of EE2, there is an agreed upon no effect level. That's basically the concentration of EE2 above which adverse effects are expected to occur. So, based on all the literature, this concentration for EE2 has been determined to be 0.1 nanograms, that's a billionth of a gram, per liter. Now you can do a mass effect calculation from this no effect level. If you think about one oral contraceptive user excretes 10 micrograms, that's millionths of a gram per day, and half of that is released in the urine into the wastewater. That's enough to pollute 50,000 liters of water, and that's per user per day. If you think about it another way, 88 kilograms of EE2 are sold per year in the US, and that's enough to pollute 1.6 times 10 to the 14th liters of water. That's enough to fill the lake behind the Hoover Dam 7 times. Now, even despite what's known about the toxicity of EE2, there are uncertainties and controversies in the literature. First is the best way to measure EE2 in the water. A lot of the methods of detection are not sensitive enough to detect it at the low enough level, near the no effect level. And that kind of leads to a dispute as to the prevalence of EE2 toxic waters. So like I said, there are definitely reports of measurements that EE2 is present in toxic levels in many effluent samples as well as surface water samples in the US and elsewhere. But the data is not robust enough to say exactly how prevalent this toxicity is among all the countries, the US and other countries as well. There's also a dispute about whether EE2 is actually causing the hermaphroditic fish. As we know, there are countless other chemicals released into the environment in modern society, and teasing out the relative contributions of each is not a simple thing. In particular, there are 3 other substances that should be considered since they may also disrupt endocrine systems. Xenoestrogens, agricultural estrogens, and human sources of steroidal estrogens. So xenoestrogens are not steroids, but since the estrogen receptor has a relatively wide pocket, other chemicals can activate it. So these are things like BPA, pesticides, PCBs, phthalates. And it's important to note that these are widely studied, and there have been reports of these chemicals causing endocrine problems in animal models, as well as some epidemiological evidence that there may be various levels of association with male endocrine problems. However, regarding the estrogen load of water, there is some reason to believe that xenoestrogens are not as large of a problem as the actual steroid chemicals. In that original isolation of the sewage water that I talked about in the beginning, when they tested the fractions of water that were activating the estrogen receptor, they did so at a much faster rate than any of the xenoestrogens they tested, and at the speed of steroids. There's also a study that looked at 35 different phthalates, and 27 of them had no effect on the estrogen receptor, and of the ones that did, the strongest one was still 1 million times weaker than natural estradiol, and that is weaker still than the synthetic EE2. There's a review article that was published looking at 71 different toxins in British surface waters, and they ranked the importance of these pollutants in 7 different ways, and found that EE2 was it made it in the top 10 of 5 of 7 of these ranking schemes. The only ones it didn't make it in were based on lethality or deadliness and environmental or governmental standards. So it's a pretty important toxin, and it ranked above all the xenoestrogens they considered as well. Another important consideration is agricultural estrogens. There is some consensus that animals excrete as much, if not more, estrogens as humans do. However, this isn't the same as saying that their endocrine disrupting potential is the same. Because first of all, animals have existed alongside humans throughout history, and EE2 is more potent and persistent than natural estrogens. There is some uncertainty, however, regarding how much EE2 might be used in livestock and aquaculture, which may obscure the role of contraceptives in the environmental problems. There is also the issue of other estrogens released into the water. Humans naturally excrete estrogens, and estrogens are prescribed for things like postmenopausal hormone replacement therapy, although much less today than in the past. But these estrogens are actually a lot more prevalent in the water. They're present in greater amounts. But EE2 is much more potent and persistent. It binds more tightly to the estrogen receptor. It's 2 times more potent than estradiol, and estradiol is 2 to 5 times more potent than estrone. But if you look at the potency in vivo, it's even higher. It is 11 to 30 times more potent in vivo than estradiol. And that's because it's more persistent in the plasma. It's harder to metabolize, and so it remains longer in the plasma, and also in the environment for that matter. It's important also to consider the bioaccumulation of a pollutant. So for something like EE2 that tends to gravitate towards the fat, it tends to concentrate in an organism, especially those higher up in the food chain. There's a bioconcentration factor that is the tissue concentration over the water concentration, and that has been reported for EE2 as being over 500. There was a study that looked at the bioaccumulation factor for EE2 versus E2, estradiol, in carp, and it showed that EE2's bioconcentration factor was much higher, and that the fish were more sensitive to that chemical. And consideration of the food chain naturally begs the question, are estrogenic environmental pollutants affecting only fish? Dr. Herrick actually searched the water quality portal, which kind of archives the water quality measurements that the USGS and EPA measure, and he was looking for whether EE2 had been measured in Kansas. And there were only 7 measurements that had been done that would detect EE2 within an order of magnitude of the no effect level. And those were done only in 2 different locations. One was at Tomahawk Creek in Overland Park area. The other was in Pittsburgh, Kansas. And you can see a map. It was in a well in Pittsburgh, Kansas. Here's a close-up of the well. And it was detected at 6.5 nanograms per liter in that well, which is over 60 times the no effect level. You can see that the location of the well, this red flag, is between a high school and a university, presumably where lots of contraceptive users spend a lot of time. So there's some question as to what surface waters drain into there and how exactly that happens, but this is certainly a concerning finding. So to summarize, EE2 is definitely present in the environment at significant levels. It's shown to affect fish in ways that are very similar to the male endocrine crisis in humans, and it's far more potent than any xeno or natural estrogen. So for those reasons, it should be given at least as much attention as other endocrine disruptors. So I'll turn it over to Dr. Herrick to wrap up.
Thank you. So far, we've discussed a lot of correlations, things that are in common between the male endocrine crisis and the environmental estrogen disruptors. Both have in common problems with fertility, problems with virility and behavior, and problems with anatomy. But correlation is not causation, right? So what can be done to investigate causation, to see if there is actually a link? And how do you do that ethically? Because you can't take human beings, particularly when they're pregnant, and give them a hormone that might not be a hormone they'd otherwise naturally be exposed to. You can't do that ethically. There are so-called natural experiments. Another word for that would be accidents in nature. And these individuals are not accidents in nature. They're made in the image of God, but they do suffer from an accident in nature called androgen insensitivity syndrome. All these individuals have a Y chromosome. They're all genetically male. And they've not taken any hormones to make them look this way. They actually have female external genitalia. And the way they get this way is that testosterone does not work in them. And this kind of phenomenon has been known a long time. It's not very common. But this, that's why I include the quote from the Gospel of Matthew. It's been known a long time. I put here on the slide 3 different accidents in nature, three different conditions that lead to a discordance. That is a difference between the genetic sex and the phenotypic sex, the sex of the body, at least the way the body looks to us. So first, androgen insensitivity syndrome, XY, male genes, female external genitalia. And the reason this happens is these individuals have testosterone. The individual in the last picture had male levels of testosterone, but their bodies are unable to respond to it, even as a fetus when you're forming your external organs. And that's because there's a mutation in the testosterone receptor. And just to review, receptors are where hormones act in the body. There's like a lock and key hypothesis where the hormone comes down, and it just happens to fit just right in this receptor. And that's why other things don't fit there. They don't have the right shape, so to speak. Where testosterone goes in these individuals, this part's broken, and the testosterone can't get in and do its thing. There's also a condition called 5-alpha reductase deficiency. These individuals are also genetically male. And when they're born, their genitalia are ambiguous. It's like they sort of look like both, but they really look more like females, even though they're genetically male. And the strange thing is that at puberty, with the surge of hormones of puberty, they actually go the way more towards their genetic sex, where their genitalia look more male-like. Yet they still have some familiar hypospadias. And this problem is due to a failure to activate testosterone. Testosterone certainly has male-type properties in the body, but it's often converted into a more active form called DHT, dihydrotestosterone. And these individuals lack that enzyme. Lastly, I put congenital adrenal hyperplasia, or CAH, and this affects both genders, both sexes. But in genetic females, in severe cases of CAH, they're born with external genitalia that look like males. And the problem there is that there's another organ besides the ovary in these affected females that's producing hormones. It's the adrenal gland, and it's got a deficient enzyme, and they end up producing, as a result, lots of male-type hormones. So why discuss these things? It's not to cause any sort of, unrest or disquiet within us, but it's to illustrate the principle of when you can't do an experiment ethically, we look in nature what has gone wrong, so to speak, and to see how that happens. And you see what they all have in common is that something's wrong with the action of a hormone in the embryo and the fetus. And this leads us to some knowledge about how things might happen with our current male endocrine crisis. So a summary of estrogens and the male endocrine crisis, observation from accidents in nature. If testosterone action is lacking in the embryo and fetus, then genetic males can form female external genitalia. And we know from other studies, not included here, that estrogens antagonize the effects of estrogen. It's kind of like a yin and yang, and testosterones have one effect, estrogens tend to undo that. If you combine these two observations, the implication is that environmental estrogens are felt responsible for the male endocrine crisis. And this is not just a conclusion of our talk. This is in all the male endocrine crisis literature, that they may not be suspecting EE2, but they're suspecting some sort of environmental estrogen. So what about behavior? Besides accidents of nature in human beings, there are also animal studies. And though animal rights activists, I think, would object to animal studies, there has been a traditional judgment in research into biomedical problems that animal studies are at least more ethical if you do them and try to, anesthetize pain, that kind of thing, and that they're ethical. So, and there have been studies done that give us insight. So, in this case, animal copulation. So, several species have a very characteristic posture in copulation. Males mount, females assume what's called lordosis. And lordosis literally means that the spine arches, and in this case, it kind of arches downward. And what these are guinea pigs and rats. And what these are doing, you see the females in front, is the female's arching her back and holding very still, and it facilitates reproduction. And the females always do lordosis, almost always, and the males almost always mount. Well, 50 years ago at the University of Kansas, W.C. Young and colleagues did a series of classic experiments that led to a hypothesis about how genitalia form and how behavior acts, particularly how behavior is formed under the influence of hormones. So what they did is they gave testosterone shots to pregnant guinea pigs. And then they looked at the effects upon the female offspring. And that was a particular interest because testosterone is not the predominant hormone in female. So they found that the copulatory position of these guinea pigs as adults would change depending on what they were exposed to as a fetus. So if you gave female guinea pigs an exposure to testosterone as a fetus, when they grew up, they were less likely to assume the typical female posture in copulation, less lordosis. Interestingly enough, if they gave the same testosterone to adult females without exposure as a fetus, no change in the copulatory posture. So this implicates a critical window of exposure in the fetus. They went on further in those studies, and they gave testosterone shots to female guinea pigs twice. So they gave it when they were a fetus, and then when they grew up, they gave them testosterone again. And they found that if they did that, not only did they do less female posture, they actually started doing male posture. Now, if they gave the testosterone as an adult only, the female guinea pigs never did a male posture. Only when they're exposed twice, as a fetus and as an adult, leading to the organization activation hypothesis. So sex hormones are thought to affect brain and behavior at two critical times, particularly as a fetus, when they organize or program the brain, kind of like a computer program. They write a program in the brain for behavior. And the same hormone, if the individual is exposed to it later in life, can activate that program, can turn the program on. Whoops. There's also been studies in humans about the hormone effects upon sexual behavior. With the androgen sensitivity syndrome, the one with the picture of the genetic males who had female phenotype, these individuals consider themselves to be female. They have female gender identity, even though they're genetic males. And certainly there's more than one factor going on here. It's not just internal workings, but external ones too, because they're born looking like females. People treat them like females until they find out at puberty there's no period. So, at any rate, there is an association of a female gender identity and a male genotype. The congenital adrenal hyperplasia is a bit less confounding in this picture. So this is the one where females can be exposed to high levels of male hormones as a fetus. As adults, these individuals have more male gender identity and increased lesbian activity compared to women who are not exposed to testosterone, male hormones in utero, pointing to a possible role of fetal hormone exposure to subsequent behaviors in adults. There's been several studies looking at finger biometrics. So the ratio, the second to third digit, is higher in men than it is in females. It's also higher in lesbians. There's more of the male type finger ratio. And the interesting thing is that ratio is thought to be related to early life hormone exposure. So summarizing gene hormones and gene discordant sexual behavior. Fetal exposure to the wrong hormone can make animals and humans act sexually discordant with their genes. And knowing that estrogen antagonizes testosterone leads to the hypothesis that perhaps the behavioral changes associated with the male endocrine crisis. Remember, not everybody considers gender dysphoria and homosexual activity as part of the male endocrine crisis, but they're happening at the same time. Perhaps these behavioral changes are due to environmental estrogens acting on fetuses. There's also been studies looking at the brain, hormones, and sexual behavior. These are slides of rat brain. So first, I would point your attention to the slide on the left, letter A. That's a male rat brain. This is a part of the brain called the hypothalamus. It's right about between my fingers here, and it's at the base of the brain. And it just so turns out that this area, these dark areas right here, you have left and right brain there, are 5 to 7 times as big in male rats as they are in female rats. Here's the same areas in the female. Okay, so you can see those dark areas are much bigger in the male. This slide is a female who was exposed to testosterone as a newborn. And you can see her areas are big like the male. So early exposure can result in brain changes that make the female internally more like the male. There's also been studies looking at sheep behavior, sheep mating behavior. And it turns out that there are animals in nature who will have what could be thought of as an analog to homosexual activity, that certain rams prefer to mount other rams. The experimental setup here is that they bunk the sheep around a rectangular bunk where they have 2 males on one side, 2 females on the other. They can't move. They have their necks in these stanchions, so to speak. And then they allow a free-ranging ram to come in the room, come in the shed, and choose who they're going to try to mate with. And it turns out that about 10% of rams will actually try to mount the other males. And they'll do that consistently. They won't mount the females. When you look at these sheep's brains, these rams, just like the rats, have a smaller preoptic area, this area in the hypothalamus, than the heterosexual males. So they have more of a female-sized preoptic area, POA. And there have been studies looking at the same thing in humans. So now we have 3 species here. Human brains are more complicated. There's actually 4 sets of cells in the preoptic area, the POA. And one of them shows some sexual dimorphism. There's a different size in the male than the female. So this is the size of the INH3, the 3rd interstitial nucleus, the anterior hypothalamus, part of the POA of humans. And you can see that males, these dots, tend to have more big POAs than the females, who tend to have smaller POAs. And they did, this is from autopsy studies, so they looked at individuals who died of AIDS, and they found, and those who died of AIDS are in the blue dots, those who did not are in the red dots, so there were other causes. But they found that the homosexual males indeed had small POAs, just like the homosexual sheep and the rats, who were female. And just like in the rats, the homosexual males had, I should say the sheep, the homosexual males had a similar size POA. It also turns out that in rats, that if you castrate a rat at birth, they end up having female-sized POA. So summarizing brain and gene discordant sexual behavior, genetically discordant POA size is associated with genetically discordant sexual behavior and can be affected by hormones. And coupling that with the observation that estrogen antagonizes testosterone leads to a hypothesis that if environmental estrogens are causing male discordant behaviors, this could be the mechanism. This is an observation from another study that shows that biology is not destiny. Okay, so there's a certain gene in fruit flies that if you activate this gene, you turn it on, you cause male fruit flies to vigorously court other males, which is something they don't usually do. Then, when you introduce another set of fruit flies to the mutated ones, these non-mutated flies, the quote homosexual fruit flies, will court the non-mutated males. When that happens, those non-mutated males change their behavior and actively participate in male-male courtship behaviors. So what you have here is a sort of social behavior that there's an increased rate of homosexual activity depending on the social interactions of fruit flies. So, in moving to our conclusion, keeping with the German theme, wie kommen wir von der Zwitterbahn runter? Or, how do we get off the Zwitterbahn, assuming we want to do that? This is a complicated question. We haven't even proven causality completely. We haven't even proven causality yet. So when you have a complex situation and you don't have causality, you can look at context. And the context of the male endocrine crisis and the environmental estrogen disruptors is that it all occurred in the context of the sexual revolution. So most of us here will be aware that 50 years ago, Paul VI made 4 predictions about widespread use of hormonal contraception: marital infidelity, lowering of moral standards, loss of reverence for women, and demographic control, all of which happened. At the same time, and Paul VI did not make any environmental predictions, but at the same time, there were others making predictions. Paul Herrick, the most famous, and they were making environmental predictions. They were predicting environmental disaster if we did not adopt the widespread use of contraception. Implicit in Paul VI's document was an observation, though, that contained the seed of a prediction. And he said that in the first place, there is a rapid increase in population. It's made many fear that the population was going to outstrip the world's resources. But he said the most remarkable development at all was in man's stupendous progress in the domination and rational organization of the forces of nature, to the point that he's endeavoring to extend this control over every aspect of his own life. One case of stupendous progress in the domination of the forces of nature was the development of EE2. The German scientists who applied for this patent in 1935 stated that the EE2 had a high physiological activity that in many cases exceeded the natural hormones. And the first clinical studies, Schering AG, the company under which this research occurred, noted the potency was so high and the potential for side effects was high enough that they decided not to market it. But they did anyway. And it has the same potency in the environment that it did in the original clinical studies. So in conclusion, there's a manifold male endocrine crisis believed to be due to environmental estrogens.
And EE2 is the most potent estrogen in the environment.
Animal and human studies provide plausible links between fetal estrogen exposure and anatomical, behavioral, gene-scored changes. So before we answer or provide preliminary suggestions for answers to how to get off the Zwitterbahn, the first thing we want to say is that in some circles, I think circles that some of us will follow public commentary, it's sort of assumed that everything we see in gender ideology, homosexual behavior is a choice. It's a problem of, if it is a problem of society, how we've been oversexed and maybe not following traditional moral standards. But if what we presented is true, there's something physical to it too, hidden beneath what we assume. And if that's the case, it's a very heavy cross to have infertility. Okay, and if you have reduced virility, reduced drive, that's a problem too. And not to mention changes in anatomy. And if you really felt like you were not what your organs appear to be, and it was something physical, actually there's a physical influence on that, that would be a very distressing thing. And if you had an appetite for your own gender that was different from most of your peers, that too would be something to be very, very hard. And I think this insight gives us in the church some reason to pause and maybe reconsider our approach to some of these problems we're seeing in contemporary society. I'm not implying at all that it changes the morality or the ethics of this problem, but it does give us pause. The other thing is to realize that biology does not equal destiny. For example, in the fruit fly experiment, when those who were not mutated started acting like those who were, it goes in both directions. Not all fetal exposures lead to gene discordant behavior. Most females who suffer from congenital adrenal hyperplasia do not have lesbian activity and do not have male gender identity. And there's also the role of free will, something that's not really subject to a scientific experiment.
And in order to limit the potential for problems from environmental exposure to contraceptive hormones, we should consider using charcoal filter on our drinking water. We should advocate for research into endocrine disruptors to further clarify sort of what is exactly causing each type of problem. And of course, don't use the pill, both for the health of yourself, your family, and for the health of the environment. Thank you.
And we're open for questions.
Okay, so you're saying the charcoal filter is going to remove the EE2?
Yeah, it should, assuming there's, it's a fresh one and there aren't so many other contaminants to overload the filter, but it's a hydrophobic molecule. It'll be.
Okay, but what about all of our fields that are being fertilized? Grow your own food. How far do you go?
Well,
you go?
yeah, and so that's another thing is that certain pesticides and things are estrogenic. So, like we said, it's not entirely clear which problem is causing what, but yeah, just.
Would you say that it's higher in cities than it is out in the rural areas, or?
Well, that's the thing is the agricultural estrogens are higher out in near farms and whatnot, but in cities you have more of the human estrogens, so it's not really been measured enough to say which would be worse.
These are preliminary suggestions. Obviously, it's a hugely complex. Took us an hour to present this data. Took us 9 months to gather it.
Right.
It's a hugely complex area, and we do not profess to have complete answers. In regard to the question about cities versus rural, I would actually answer a separate question, country versus country. So a lot of the male endocrine crisis data was in the UK and Denmark. And I don't have knowledge about Denmark waters, but UK does not have near the amount of fresh water that the United States does. And they have a higher population density. So there may be a bigger problem in the UK. There's some data to suggest that. There's some data in the US that suggests that maybe it's not as bad. Something like 12%, don't quote me on the figure, of the US is of the world's fresh water is in the Great Lakes in the United States. And we just have a bunch of fresh water in this country. On the other hand, that well in Pittsburgh is 1,000 feet deep. So there is an organization in the Ozarks who puts fluorescent dye into surface water and then pumps up groundwater. Groundwater is below the ground in the aquifers. And they have measured hundreds of gallons, I'll say per hour, leaving the surface and going down into the groundwater through sinkholes, and then there's something like sinkholes in rivers where you don't see it, but hot water's going down. So that could be a putative way how it gets from the surface, where you're getting a man-made chemical 1,000 feet in the ground. That's all I got. That's another thought I forgot. So, anyway.
So you had noted that in the fish studies that it built up in the soil as well. It seemed. Do we know that it does it break down? I mean, how fast does it break down?
Not nearly as fast as a natural estrogen, and yet, since it does tend to gravitate towards the organic material, it will accumulate in the sediment and then kind of redissolve in kind of a cycle like that. So, yeah, there is some evidence. There were measurements of EE2 in the sediment and in the water, and EE2 is usually higher than other sorts of estrogens in that component.
I remember what else I was going to say. The Ozark Aquifer is where they have that well in Pittsburgh. It goes down into the Ozark Aquifer. That covers virtually the entire southern half of the state of Missouri.
And are you saying that the EE2 is also given to the animals? They're using that same estrogen
So-
with your lifestyle?
That's what, in the many papers we read, there was one study that said, what was it, a personal communication with someone said that it's not
Veterinarians.
used, right.
Veterinarians. and animal
Veterinarians,
control
it's not
experts,
used
yeah.
in animals. But then another paper said that it is, and they had more credible sources, and so we think that it is, but we haven't found numbers on exactly how prevalent that use is, so that's why I put that caveat in there, that, you know, if it is, obviously it should stop, but that could be obscuring what we're seeing with human excretion of this chemical.
Apparently, aquaculture is when you grow fish, you know, like your farm fish. And apparently, it's a desirable thing to have all your fish the same gender. So I grew up on a farm, and what we would routinely do is neuter all the males, because males get aggressive and they hurt the other pigs, and that tends to have them make less meat, you know. So it hurts your commodity. There's some sort of similar feature in fish, and so they'll actually apparently give, according to this one source, Harrick and colleagues, they'll give ethinyl estradiol to farm fish to make them all the same gender on purpose. And they tend to dump their waste right out from the surface. So, probably another avenue for changing action that seems to be the least necessary of all these uses of the hormone.
When we say a charcoal filter, is that like a Berkey or a Brita?
Yeah. And reverse osmosis would usually have charcoal filter built in too, but yeah. Right.
Yeah,
As far
and
as
again,
I'm aware.
preliminary, this is far from settled, but it's probably something we can do on an individual basis. Since we started reading this, I started getting all my water out of our fridge. Of course, I'm not an embryo anymore.
I started wishing I knew all this before I had sons.
One thought is that if ethinyl estradiol goes to fat more than water, and everybody has fat in their body, so if we're worried about the environment, the embryo has an immediate environment. It's the woman. So if she had taken, as far as I know, no one's done blood levels on women after they quit the pill. But what's their blood level? You know, we've been doing this for 50 years. Thank you.